All right, everyone. I'm going to go ahead and get started. My name is Professor Dana Babayu. It is my pleasure to introduce you to the cell molecular bio class for engineers. I had the pleasure of taking this class about 16 years ago. So if any of you wonder where you're going to be in 16 years, hopefully you guys can say not in this very same room. I know I go for self-deprecating humor, forgive me. I made an announcement, I posted an announcement, but for the sake of trying to fit all of the content and not have us too hurried over the course of the semester, instead of having the introductory lecture I usually give today, we're going to go ahead and jump into content today. But again, I posted the lecture that has a lot of the information about what the semester is going to look like. I'm going to provide a few quick details before we get started today. But before we jump into some of the introductory stuff, I also want to introduce some of our TAs. We have Timothy Ho in the back. Timothy, can you say hi? He's our graduate TA for this course. He's a fourth year grad student in our program here. We also have Cooper, who was an undergraduate TA for me last year. So he's a seasoned vet and is going to be a really great resource for you all. The rest of our teaching team, our undergraduate TA teaching team is still being rounded out, but I'll make sure that I properly introduce the remainder of our undergraduate TAs. The way the class is going to work is that we're going to have content that we discuss and apply it to some sort of disease or application point. And so we'll jump into talk about the background for whatever that disease is, and then jump into whatever the content for that lecture is. For example, today we're going to be talking about amino acids and proteins and applying that to sickle cell disease. And it's going to be that same format throughout the course of the semester. And so primarily, or I should say, especially given that usually at this point, about a third of the students in this class are pre-med, it's a great opportunity to see how what we're talking about applies to some of the clinical and medical problems you guys will be facing. And for those of you who are going into research and development, this is also an opportunity to see where there's opportunities or angles for new avenues for research. There's also going to be an opportunity at the end of each lecture for me to highlight some of the people within our research community at UVA who are studying these topics or studying these ideas and helping to identify some of those people that you guys can maybe get in touch with to try and get started and plugged into a research lab. For every couple of lectures, we're going to have a quiz. And then with each quarter or unit, we're going to have an exam. So for example, and I should say every Thursday we'll have a quiz. So for example, this Thursday we'll have a quiz on this content. The quizzes are really short. They're going to be 7 questions. And it's meant to try and help give you an opportunity to work towards applying the content and understanding it well. And there's going to be about 15 quizzes throughout the course of the semester. So if you don't do well on a quiz, that's okay, because there are a lot of them. The way this is also going to work is that there's going to be 4 exams. There will not be a cumulative final. There will just be a fourth final. There will be a 4th exam that will be during our final period. And for each quarter of the class is broken into quarters. If you end up taking the quizzes and the exams, whichever grade is better will be your grade for that quarter to kind of help you keep pace. So you can decide and determine I'm only going to do quizzes or I'm only going to do exams, or you can do both and decide whichever one's better, that'll be my grade for that quarter. There's also going to be a semester-long project that you guys will be working on, but the information on that is both in the syllabus and goes into further detail in the recorded lecture that I provided for you guys. Another thing I'm going to say is that I'm going to have multiple points at which I stop to provide room for questions, also for me to get a sip of water. But I love for you guys to ask questions. A lot of times, if there's a burning question that's in your mind that you're afraid to ask, the chances are there's someone else in the crowd that has the same question in their mind. And if you're afraid that the question you're asking is a dumb question, feel free to ask it. It makes me feel good because it's easy to answer. But that also might mean that there's a fundamental thing that I did not do a good job explaining. So if there's questions that you guys have that if there's something that didn't make sense or I didn't do a good job explaining, that's on me. So with that being said, please feel free to ask questions either during a point at which I stop for questions, or a point at which, or at the end of any slide. With that being said, I'm going to stop for water and ask, are there any questions on anything I touched on? Oh, and one last thing I should say. Sorry, I just remember this. A fair number of students are going to be hiking over here from Thornton. And so today I wanted to try and make sure that I touched on some of this introductory material that, again, is covered in great detail in the introductory lecture I provided. But given that students are hiking over from Thornton, um, I will probably usually try and give a few extra minutes, definitely when we have quizzes on Thursdays, but or quizzes and exams. But for lectures, I'll also try and give a couple minutes for people to make their way over here. With that all being said, are there any questions there? All right. If there's no questions, we'll go ahead and jump into the content for today. So, oh, I guess one last thing I forgot to mention is that all lectures will be recorded. And so I'm going to post them on Canvas. On the homepage, there's a kind of a table of a course calendar with links for the pre-lecture videos. So there's going to be some for about a half to two-thirds of our lectures. There's going to be like a brief 20-minute video to help create a common foundation because everyone's coming in with different degrees of knowledge here. And so the pre-lectures help create a common foundation for us to be able to go into detail and to the depth that we need to for the lectures. Um, and I'll record and provide a link for the recorded lectures because there's going to be often times where students have multiple commitments. There might be a career fair, or there might be, for some of you who are in research, you might have a long experiment that's taking your time, or some of you might be sick or out of town, especially for those of you that are athletes and have to travel. So lectures will always be recorded and provided on Canvas. With that being said, you benefit a ton from coming and being present in class. As well as there's going to be office hours that we're going to be organizing throughout the course of the week hosted by the teaching team. I've already posted mine online. Mine are going to be Mondays and Wednesdays at 3 p.m. in my office. Uh, it is very easy for me to say this confidently. Um, those office hours are meant to be a resource to you guys, and I highly recommend you come to office hours. I am so happy when there's, instead of 2 or 3 people, if there's like 10 or 12 people, and it becomes an opportunity for you guys, for us to talk about the content and for you to enhance your understanding and talk through this material. Um, there's a couple exercises that I posted on that pre-recorded lecture that help understand that going from first year engineering curriculum to second year BME curriculum is a big step because first year curriculum, not to diminish it and not trying to be reductive, it's a lot of memorization. Second year, especially in this class, is not just memorization, but it's going to be a lot of understanding and application. And so that's the way in which the questions on the quizzes and exams are designed. And being able to talk through the material is a really helpful way to understand and apply the material so that you're well prepared when we go through our quizzes and exams and even for your projects. And so I highly recommend going to office hours. I can say with fairly high confidence that of the students who would regularly attend my office hours, if they attended about like 90% of them, each of those students probably got either an A or an A plus in this class. Not because I gave it to them as a reward, but rather it was a useful resource for them to help understand the material. And so now that I have a sample size of about like 4 or 5 years now, it's become abundantly clear that those who come to office hours do much better in the course. And so I highly recommend you guys come to office hours. Again, we're going to try and iron out the details for our office hours and make sure it's available throughout the course of the week so that it is so that we are as accessible to you guys as possible. Okay, I'm going to stop. I'm going to switch to the content now. So today we'll be talking. We're going to be focusing on discussing what about amino acids and proteins and applying them and trying to understand the structure and function and applying them to sickle cell disease. We're also going to be talking about what are the evolutionary constraints that determine amino acids and protein structure. And we're going to be applying all of this to sickle cell disease and understanding why amino acids and proteins, how they relate to the etiology of sickle cell disease. There's going to be throughout the course of this semester, there's going to be words that are highlighted in blue that are important words for you to know. Not that you will be assessed on some of them, but it's meant to try and highlight what are important things for you guys to know here. So etiology usually refers to the cause of a disease. So sickle cell disease is a recessive inherited blood disorder. Recessive means that you need both copies of this mutated of this mutated version of the gene, um, there's always two copies of every gene that we have. And so you might hear of both recessive and dominant. In the case of a recessive disorder or recessive disease, that means you need both copies, both alleles. You'll hear alleles from time to time. That just means the copies, both alleles of this disease mutation. Uh, and it's caused by one single mutation in the hemoglobin beta chain gene. So whenever there's a disease that is caused by one mutation, that's called a monogenic disease or monogenic disorder. A very small population of diseases are monogenic. Sickle cell disease is one of them. Cystic fibrosis is another. Most diseases are polygenic. Polygenic meaning that there's multiple genes that are responsible for this disease. So sickle cell disease is monogenic and recessive, and it's been pinpointed down to the beta chain gene of hemoglobin. The gene name or the gene shorthand for the beta chain, hemoglobin beta chain, is highlighted in blue. Not to say that you need to memorize the gene shorthand, but rather to point out how we write the shorthand for gene names. So for human genes, it's all caps italicized. And so if you see that, that's going to indicate to you immediately that this is a gene that we're talking about. So some of the symptoms associated with sickle cell disease is that the erythrocytes or red blood cells are sickled. So you can see here in this image that the red blood cell that has HB for hemoglobin, AA means the normal allele, so it has both copies of the normal alleles, is usually kind of like a donut or bagel shape. And that's really helpful for these red blood cells to be able to squeeze through tiny capillaries and get to various parts of our body. But the red blood cells that have hemoglobin with the sickled, both copies of the sickled allele, you see that here with HBSS, has this sickled-like shape. And some of the issues that can emerge with that is that what ends up happening is that it's not pliable like a normal red blood cell, or the red blood cells that have sickled hemoglobin will end up stacking on top of each other, and they're not pliable, and they can create occlusions within some of these capillaries, these small blood vessels. And so what ends up happening sometimes is that as these red blood cells stack on top of each other, they can cause red blood cells to lyse or burst open. And so anemia is the loss of red blood cells. And so there's also sometimes a lot of pain associated with sickle cell disease. So there's acute chest syndrome. There's a lot of chest pain associated with it. And some of you may already know this, that iron is an important component of our red blood cells' ability to carry oxygen to various parts of our body. So when these red blood cells lyse, they end up releasing a lot of iron. This iron is an important factor in bacterial growth, and so the release of iron can actually be a contributing factor to bacterial infections that occur within some sickle cell patients. And then finally, a final symptom is dactylitis. That's going to be inflammation of the digits. And so they're going to present with swollen fingers and swollen toes as well. We're also going to typically talk about the prevalence versus incidence of the disease. Prevalence usually describes what is the current state of the disease. So current state of sickle cell disease is that about 90 to 100,000 cases present within or have been reported within the US. When we talk about incidence or incidence rate, that's a projection in the future of how many patients will have the disease. And so the incidence rate, excuse me, is about 1 in 500 African American births. We'll talk about towards the end of the lecture why it presents primarily within African American populations. But that's the incidence rate and prevalence of sickle cell disease. Before we jump into the peptides and amino acids portion of the lecture, are there any questions on sickle cell disease here? Can you repeat that again? Uh, treatments will be important to know, and that's something we'll talk about at the end of the lecture. At least the treatments that we talk about. Yes. It's referred to as sickle cell anemia when there's anemia that presents within the patient. All right, so we're going to spend some time talking about peptides and amino acids, and then we're going to pivot back to sickle cell disease and describe how it relates to the etiology of the disease. Um, So peptides are short fragments of proteins. And what's important to know is that amino acids are the building blocks of proteins. Amino acids are if you think of, I'll refer back to Legos a lot when talking about amino acids and proteins. Amino acids are each of the individual Legos, and the protein is the completed structure of Legos that create something. Um, and so a peptide is a linear chain of amino acids that are strung together, and they're usually held together by a specific kind of bond called amide bonds. And so usually a single amino acid is going to be a single amine here that goes to, some of you may already know this, but we're going to have, wherever there's a junction here or here, that's going to be a location of a carbon. So this, off of this amine, we refer to this as the alpha carbon. From there, there's something called a side group here. From there, we have this oxygen that goes down to another amine on an adjacent amino acid. But this is like the functional unit of what an amino acid is. So we have the carboxyl end and the amine end. And so we have an alpha carbon here and the beta carbon right here. Each amino acid is going to have this same structure where we have an amine and we have a carboxyl group here. But the specificity of each amino acid is determined by this side group. And so whenever there's a general, and I can also draw it over here because I imagine for some of you on this side it might be hard to see. So the alpha carbon here, and this is referred to as the beta carbon. So the specificity of every amino acid is at this side group. And when you have multiple amino acids together, they're bound together by an amide bond. And so where the amine is, as we string them together, there's a directionality for these peptides when multiple amino acids are strung together. The way we determine this directionality is important. And so there's a couple different ways you can go about determining it, but we're always moving from the N terminus to the C terminus. So if we had a string of amino acids together, looking at this one here, we're always moving from the N terminus, this left side is the N terminus, all the way down to the C terminus. As you can imagine, N corresponds to amine, C corresponds to carboxyl. And so what's important to know is that, you can imagine that if this marker was a peptide, it could be flipped around. Protein biology is in three-dimensional space, so it could be flipped around in any orientation. So it's important to know what's the directionality of this protein or this peptide. And so to help determine the directionality where the N terminus and the C terminus is, there's a couple of ways that you can go about doing this. The simplest way that I've found is that you just go smack dab in the middle of this peptide at an amine that's in the middle, and you look to the left and to the right. If you look to the right, there's an alpha carbon here, and we know it's an alpha carbon because it has a side group. And if we look over here, this is a beta carbon because it has the double bound oxygen that corresponds to a carboxyl of an adjacent amino acid. So that means, sorry, I lost my mouse. So if we look to the right, we can see, okay, this is the alpha carbon with a side group here. So we're moving towards the right here because we're moving towards this carboxyl group here. And so if we were going to continue stringing amino acids here, this is a single amino acid. It would be bound to an amide of another amino acid here, and this would be the N terminus, and this would be the C terminus. Again, this directionality is determined by looking at uh, by trying to determine where the N terminus and C terminus is. And one way you can do that, again, is looking at an internal amine here, looking to the right and to the left, and you want to move towards, this is pretty reductive, but it help if you find the amine, if you look for the adjacent alpha carbon that has the side group, that'll tell you which direction the C terminus is. Because you're kind of looking within that single amino acid and you're finding that alpha carbon and realizing, okay, this is the direction we're moving in when reading these peptides. The specific kind of amide bond, amide bonds are a class of bonds. Of those, the ones that we find here that join together adjacent amino acids are called peptide bonds. And so they're because of the resonance frequency, they provide a rigid and planar quality to them. But it's these peptide bonds that help string together multiple amino acids together that create a peptide string. So two important qualities of protein sequences is that they're both conserved and customized over the course of evolution. The way we'll talk about in a moment, the way in which they're conserved and customized. When we talk about proteins, there's multiple degrees of structure when we talk about them. So the first is primary structure. Primary structure is simply just listing out the amino acid sequence, listing out the Lego pieces that are involved with creating this protein. And for every protein, there's a gene that corresponds to it. And so the amino acid sequence is determined by that gene. So the primary structure, again, is just listing out in order the amino acid sequence of that protein. And so when we look at beta-chain hemoglobin, and we look at its primary structure across multiple species, these species being this important, it's not critical for you guys to know, but we have up top humans, and we have macaque monkeys, cow, sheep, boar, bat, mouse, chicken, and crocodile. If you just take a moment to look at the primary structure here, looking at the amino acid sequence, there's a fairly clear similarity of this gene of this amino acid sequence across all of these species. So this is the conservation of protein sequences across species, across evolution, and this is across about 160 million years of evolution. And so, what's also important to note is that there's sometimes going to be small changes that you see. You might be able to identify specific amino acids that might change from one to the other, but the segments that are conserved usually there's a reason or rationale or justification for their conservation. And so it's going to be important to sometimes be mindful of that. When we talk about the similarity or degree of similarity across these species, we usually refer to that as homology, especially in research when we're comparing human versus mouse genes or human versus mouse proteins. We ask how much homology is there between these proteins. Now, there's also customization. And so we have, whenever there's a customized version or flavor of a protein, that's usually referred to as an isoform. And so different isoforms will be encoded by different genes. And so here we have, moving from top to bottom, we have the beta chain of hemoglobin, the delta chain, the epsilon chain, and the alpha chain. It's believed that there was originally one ancestral gene for hemoglobin, and that there was a mutation that caused two copies to occur during replication. It was selected for, it's believed that it was selected for over time that it was favorable to have those two copies. And then there was another mutation replication event that caused it to go from 2 to 4. And it was, again, advantageous to have 4 copies. This is what's theorized. But that's how we can develop, that's how over time, over evolution, there's different isoforms that emerge. And so whenever you hear that there's a different isoform of some protein, that's usually a totally different version or flavor of that protein that's encoded by a totally separate gene here. So this is a table of the different amino acids that we have in nature. Um, I usually like to crack the joke that you have to memorize all of this, but it usually doesn't get very many laughs, so I'm not going to make that joke here. I'm not asking you to memorize this entire table. I will, throughout the course of this lecture, try and identify specific amino acids that I want you guys to know. Um, when you take for those of you who take biochemistry, you will have to memorize this, but I'm not going to ask you to do that here. Um, and so again, I'll highlight and identify what are the important amino acids that I want you guys to know here. I know you probably didn't find it very funny when you were a student in the class, Cooper. Um, so one thing that I'm gonna keep coming back to is that there's a relationship, and this is particularly true as engineers, that structure determines function. There's a relationship between structure and function, and that is particularly true with proteins. And so when we go from a linear chain string of amino acids, how do we go from this polypeptide chain, into a three-dimensional folded protein? Um, and, and there's going to be multiple ways in which we're going to see these proteins, multiple ways in which we see these proteins illustrated. But we're going to be talking about now how the qualities of some of these amino acids and some of the key bonds that determine the folding of proteins and the bringing together of multiple proteins to create a multimeric protein structure. Before we move on, I'm going to I'll stop for two or three questions here. And again, I love questions. Highly recommend them. Yes. Yes. So there's going to be about 4 to 5 key groups, and it's going to be fairly intuitive when as we talk about it further, because there's going to be um, almost pairings of groups, positively charged, negatively charged, hydrophobic. And so it will be important really for the application of this content to know those groups, but I'm not gonna ask you to memorize each member of that group. If that makes sense. Yes. So an amide bond is defined by what specific atoms are associated with this bond. And a peptide bond is a certain kind of amide bond. But it's, what's the right way to say this? The participants of the amide bond are satisfied by the amine group of one amino acid and the carboxyl group of the adjacent amino acid. And so a peptide bond is a kind of amide bond, but uh, the necessary participants to constitute an amide bond are just satisfied by what is part of a peptide bond, if that makes sense. So it's just a kind of amide bond, that's all. A peptide bond is a kind of amide bond. All right, so the first category of amino acids we're going to talk about are those that have, we define them as having special side chains. And so the first one that is going to be important for you to know is cysteine. When we're talking about some of these amino acids, it will be important for you to know the 3-letter abbreviation. I'm not going to ask you to know the single letter abbreviation. So what makes cysteine noteworthy is that it's a sulfur-containing amino acid, and that the side group here is constituted by this sulfohydryl group. We usually refer to that as a thiol. And so this thiol right here is going to be important for modifications to proteins. Another one that's important to know is proline. What's really unique about this one, as you can tell by its structure, is that the side group bends back around to the amine here. And so this structural property of proline can actually induce kinks in a protein. So for example, one of the most abundant proteins in our body is collagen. That's made up of a lot of proline. And because of that, it has a helical structure where there's constantly kinking around that determines the structure of collagen. And so the key thing to note here about proline is that it has this unique structure, and that for cysteine it possesses this thiol within its side chain here. And so that's going to be important to know because one of the important bonds that emerges within proteins are disulfide bonds. The way in which that happens is that there's a bond that emerges between two thiols on different cysteines that creates a disulfide bridge across those cysteines. And so typically what ends up happening is that emerges within specific environments. When we're in a reducing environment, we're not going to have that disulfide bond occur. And there's going to be particular explanations as to why that's favorable. But when we're in an oxidizing environment where we're losing electrons here, there's going to be this disulfide bond that occurs. And so typically, these cysteines will as an example of the environments in which that'll happen, these cysteines will remain reduced and not create a disulfide bond in environments like the cytoplasm. So as proteins are being synthesized, and if they're being processed out into the cytoplasm, they're not ready typically not ready for their bioactive shape. And so the cytoplasm is a reducing environment where we're not going to have those disulfide bonds occurring quite yet. However, in oxidizing environments, like the extracellular space, or within some of the organelles within a cell, like the Golgi or like the ER, they need to be in their bioactive shape, ready to carry out their function. And so those environments are typically oxidizing and is an environment where that disulfide bond will occur. One example of an instance where disulfide bonds occur is within insulin. So we have two separate polypeptide chains that constitute insulin. We have chain A and chain B, and there's 3 disulfide bonds that occur. We have one that occurs across the alpha chain itself, one that occurs between these two cysteines here between A and B, chain A and chain B, and then over to the right between chain A and chain B here. What's important is that without these disulfide bonds, insulin would not be in its bioactive form. So these bonds are critical for different proteins to actually carry out their function. Another form of bond is ionic bonds. It's a strong electrostatic interaction that involves the transferring of electrons between atoms. And we typically, in a research setting, we'll typically try, when we're studying a specific protein or trying to analyze proteins within some sort of given sample, we'll actually be able to use salts to try and break these ionic bonds to disrupt this electrostatic interaction and be able to separate them and what's the right way to say this? And be able to we'll be able to disrupt these ionic bonds and have these proteins in a more linear shape so that they're more accessible, for example, to antibodies, so we can detect them and study them. So the next group of amino acids are positively charged or basic amino acids. We have arginine, lysine, and histidine. I would say arginine and lysine are particularly important for you guys to know. We'll be talking about positively charged amino acids, and we'll be exploring the consequences of substituting out positively charged amino acids. And so it's important to know which ones are the positively charged ones. Histidine is positively charged within a specific pH, so it's kind of context dependent, the way in which it acts. But the ones that are usually positively charged or basic are going to be arginine and lysine. The acidic and negatively charged amino acids are aspartate and glutamate. Depending on their state, you might hear them referred to as aspartic acid and glutamic acid. But these are the negatively charged amino acids. And again, I should have taken a step back to describe this. But again, the side group is what determines what the amino acid is. And we look to the side group to see whether or not it has a charge. So if I go back and look at these positively charged ones, we can see that there's a positive charge on this side group here and on this side group here. when we look at aspartate and glutamate, there's a negative charge on the side group here and a negative charge on the side group here. And so it's important to note again, it's important here for you guys to know what are our positively charged and negatively charged amino acids. So actually, I'm going to stop there and give room for a couple questions. Yes. So I think there's what I hear are two questions in there. I'm not asking you to memorize the structures. Again, save that for biochem. You guys will have to do that then. I'm not trying to terrify you guys with biochem. I was terrified of it when in undergrad, so I think that's why I keep saying that. Not asking you guys to memorize the structures. I am asking you to be able to identify, if you're given the three-letter abbreviation, what amino acid is that? Yeah. I want to give space for another question or two here. Yeah. Hold that thought. We're going to talk about that on this slide. Great question. Yeah. Yeah. So the environment in which these proteins can be found throughout our body, pH can change dramatically in certain circumstances, and within some biologically found pHs, histidine's charge can change. And we'll actually be talking about that in about 5 or 6 slides about how pH relates to identifying like the charge or the electrostatic qualities of a protein. But histidine just has a unique quality where it's kind of context dependent how positively charged it is. All right, so there was a question a moment ago about ionic bonds in these proteins. So when one thing to note about hemoglobin is that we talked about beta chain. We had a slide where we talked about how there's a beta chain and alpha chain that we have in biology. Those are building blocks that make up the hemoglobin that we have within our red blood cells. And so hemoglobin is a multimeric protein structure. So there's what makes up hemoglobin is 2 alpha chains and 2 beta chains. So there's 4 subunits together that make up the hemoglobin multimeric structure. So going back to the Lego analogy, if you had a fully compiled beta chain, if you had 2 fully compiled beta chain Legos that are the full protein, and 2 alpha subunit of hemoglobin, those 4 parts together make up hemoglobin. So whenever we talk about proteins that are made up of 4 parts, that's called a tetramer. If it's 3 parts, it's a trimer. If it's 2 parts, it's a dimer. So hemoglobin is a tetramer. Because it's a mixture of 2 different kinds, we call it a heterotetramer. So hemoglobin is a heterotetramer made up of 2 alpha chains, or 2 alpha subunits, and 2 beta subunits. When we look at, when we're talking about the structure of proteins, looking at the primary structure, which again was just listing out the amino acid sequence, looking at an individual subunit or an individual protein, looking at a structure like this, that's called its quaternary structure, or tertiary structure. The tertiary structure is looking at one specific protein. In the case of fully assembled hemoglobin, this macromolecular structure, when we bring the 2 alpha subunits and 2 beta subunits all together, we refer to that as the quaternary structure. Okay, so just to kind of bring it over here, a single individual protein, looking at its folded structure, that's referred to as its tertiary structure, the 3rd degree. When we have a protein, not all proteins create some sort of multimeric structure. The instances in which multiple proteins come together to create a large multimeric structure, when we look at its folded structure like this, that's referred to as its quaternary structure. Okay? And so again, hemoglobin constitutes two beta subunits and two alpha subunits. Um, the red that you see here is just the protoporphyrin ring where iron is bound. But taking a step back to the ionic bond question earlier, um, if we look and mark the positively charged and negatively charged amino acids, marking uh, marking lysine, arginine in red and glutamate and aspartate in blue, if you take hemoglobin and break it in half, what you can actually see is that the negatively charged and positively charged amino acids kind of correspond to each other when you pull them apart and mark them. And so at the quaternary structure level, ionic bonds and these electrostatic interactions are what drive the bringing together of these multiple subunits and keeping them together. Um, and so we'll talk about towards the end of the lecture what happens if you disrupt that. Um, yeah, so you can appreciate that they're complementary and that the positively charged and negatively charged correspond to each other when you actually pull apart hemoglobin here. So we talked a moment ago, and there was a question about changing of pH and how that can alter histidine structure, or sorry, histidine's charge. Forgive me. Um, there's an actual property of every protein that's unique to them, that helps us understand how positively charged or negatively charged they are. So if we had a tennis ball protein, for lack of a better phrase, and if we had this conduit that was a gradient of pHs, um, and if we put it within this gradient, when we're at the lower pHs, where it's more acidic and there's an abundance of hydrogen ions, what's going to end up happening here is that negatively charged amino acids will take up those hydrogen ions, and it'll help us understand how how negatively charged it is there. If we move it down to a higher pH, where there is a lack of hydrogen ions, the positively charged ones will end up donating there. And so what we're trying to do here is this will help us determine the electrostatics or the charge of a certain protein. Because every protein has a unique amino acid sequence, it has a unique balance of charges to it. And so what ends up happening is that there's an actual experiment where you can take proteins and run them through a gradient of pHs, and the pH at which it's electrostatically neutral, where all of the positively charged amino acids have been donating their hydrogen ions, and all of their negatively charged amino acids are taking up hydrogen ions, there's a balance point at which it's perfectly electrostatically neutral. The pH at which that happens, where it settles, is called the isoelectric point. Again, the pH at which a protein is electrostatically neutral is referred to as its isoelectric point. And every protein has a unique isoelectric point. And that helps us understand how positively charged or negatively charged it is. So we've talked about primary structure. We've talked about tertiary and quaternary structure. Um, Secondary structure is a quality of these proteins that is primarily driven by that is determined by folding driven by hydrogen bonds. So these hydrogen bonds are typically going to be it's going to be an attraction that occurs between some sort of electronegative atom in one molecule and a hydrogen from another, primarily those bound to another electronegative atom. And so it involves some sharing of electrons, but the simplest example of this is water. And so we have hydrogen bonds that occur between each water molecule, where the hydrogen atom on one water molecule is going to have a hydrogen bond that occurs with the oxygen on a totally separate water molecule. And so to help us understand this as it relates to amino acids, the hydrogen present on the hydrogen present on this amine on one amino acid over here is going to have a hydrogen bond with this oxygen coming from a totally separate amino acid here. And so when we talk about that, that hydrogen bonds determine the local folding, what this looks like is there's going to be one type of secondary structure where there's going to be hydrogen bonds that occur vertically from one amino acid to the next. And because it's all occurring vertically, what ends up happening is that it creates a structure where all of the side groups are pointing outwards. This is called an alpha helix, okay? So a key thing to the two qualities of an alpha helix here that are important for you guys to know is that all of the side groups are pointed outwards and the hydrogen bonds are occurring vertically here. And it creates this alpha helix structure. The next structure is called a, oh, sorry, forgive me. And so in hemoglobin, if we take a second to look at the secondary structure there, it is almost entirely comprised of alpha helices. The second structure is called a beta sheet, where if you look for a moment, the hydrogen bonds that are denoted by the dotted lines are occurring not necessarily vertically, but across from one side to the next. And what ends up happening is that the side groups are alternating their orientation from one amino acid to the next. And so what ends up happening is that the structure kind of resembles kind of a corrugated aluminum ceiling, if you guys have seen those on some sheds. And so we call this an anti-parallel because these peptide strings are going in opposite directions. It's an anti-parallel beta sheet. There are instances, this is typically what happens, there are rare instances where there are parallel beta sheets, but that's fairly rare. You can see here that adjacent to this alpha helix here, that there are parallel beta sheets, but typically we see them occurring in an anti-parallel fashion. I will typically I'll typically ask to do an I'll typically do an exercise here where I'll ask you all to stop and determine like what the directionality is here of the peptide. The arrows kind of give it away, and I keep trying to tell students to ignore it, but it doesn't really work. But again, if we take a second to look at these peptides, again, the anti-parallel, they're going in opposite directions, which means they're going to have different directionality. So if we wanted to determine where the N terminus is and where the C terminus is, again, we're always moving towards the C terminus. You can think, it's always helpful to have some sort of like abbreviation or shorthand to remember. We're moving towards the C terminus because that's where it closes, for lack of a better phrase. And so if we wanted to figure out the orientation, say, of this bottom peptide, we could look smack dab in the middle, find the nitrogen that's part of the amine, and we can look to the left and look to the right. If I look to the left, I find the alpha carbon, and I know it's the alpha carbon because it has this side group here. And if I look to the right, this carbon has the double bound oxygen, which means this is the carboxyl of an adjacent amino acid. And so the directionality, the C terminus is going from the amine to the alpha carbon that has the side group here. So I know that this is the direction of the C terminus. If I look to the top, I wish I was about a few taller. If I look to the top, again, if I go smack dab in the middle, I'll just find an amine, I'll look at the nitrogen, I'll look to the left, I'll look to the right. If I look to the left, there's a carbon that has a double bound oxygen, which means it's part of the carboxyl of an adjacent amino acid. If I look to the right, there's a carbon that has a side group bound to it. So I know that's the direction of the C terminus. Does that make sense? If it does not make sense, well, sorry, I shouldn't ask that. I'll describe it really briefly. To determine your direction, you always want to try and find you will always be able to move. You'll always be able to determine where the C terminus is whenever we're trying to find the direction of a peptide by looking at the amine, looking at the nitrogen, and finding the alpha carbon with a side chain on it. So if you look at the nitrogen, look for the alpha carbon that has a side chain on it. Now, when we have a peptide string that doesn't have the shorthand for just side chain, but has actual side groups where there might be the thiol from a cysteine, there might be the proline king structure. It's going to be a little difficult to try and identify what the alpha carbon is. If you look to the left and right, if one of them is has a double bound oxygen, you know that's not the alpha carbon with the side chain. That means the other direction is the alpha carbon with the side group. It's usually easy to tell if it has particularly a really complicated side group, like some of them do. But you're always going from the alpha carbon. That's the direction of the C terminus. You want to move in that direction. Whenever we're talking about not just proteins, but even genes, all of them have directionality. And so understanding what direction the C terminus is in is also important when we take a step back and understand how proteins are made when we get to that unit later on in the semester. I'll stop there and give space for a couple of questions. Yes. Yeah, so for the sake of the recording, can you have alpha carbons and beta sheets within the same peptide string? Yes. And that's going to be determined typically by the usually determined by the side groups. We're gonna have a really helpful example of that next class. But yes, within a single polypeptide, yes, you can have uh, different secondary structures within a single polypeptide. I'm gonna go here, then there. Yes. Yeah, that was really similar to the previous question. So it's typically going to be what the side chains are of those amino acids. Um, so to help perhaps maybe illustrate the example, um, the I'll take a couple slides back. So these side chains are pointing outwards. Um, and again, structure impacts function. There are certain regions of our certain parts of our cell that are fairly hydrophobic. And so if these, and we'll talk about hydrophobicity or hydrophobic in the next slide I'm going to discuss, but if all of these side chains are pointing outwards, what's going to end up happening is that this is going to be a protein that is designed to be in a hydrophobic environment. And so it's going to be typically creating an alpha helix structure. And so if there's sometimes going to be a mixture of different, it's not necessarily always going to be only hydrophobic here. For example, we just looked at the structure of hemoglobin, and it's all alpha helices. They're not all going to be hydrophobic. We looked at the positively charged and negatively charged amino acids a moment ago, and they're also present within it. And so there's what I should also say is that there's a lot of what we believe. I'm going to take one more step back. This is a class of half-truths. There's only so much we know at this point about some of the things that we discuss. For example, understanding how amino acid sequence relates to the structure of a protein is an entire field of research called structural biology. Just this past year, the Nobel Prize went to AlphaFold for helping us understand how primary structure individual amino acids can help us understand the structure of a protein. So far what we know is that the orientation of these hydrogen bonds and the orientation of these side groups here, the individual amino acids and helping us understand how they might fold in space and how certain bonds might occur will help us determine, is this an alpha helix? It is really hard to determine what the structure of a protein is going to be, which is why usually when someone wins the Nobel Prize for something, it's something they did 30 years ago. AlphaFold was created in 2018, and they won the Nobel 6 years later because this is such a big deal to help us understand proteins that we don't know a lot about so that we might be able to create new drugs. But why certain structures are, how structures are, what we know so far is that it seems to be at the amino acid level. and that the orientation and ordering of those amino acids can determine uh, how the secondary structure is created. It's the best answer I can give at the moment. Forgive me. I think you had a question, and then we're going to keep going. Yeah, so I'm going to use these two markers as an example. So each of these is a single protein. We're looking at them in their protein structure as depicted. I'm going to move forward here. So this is looking at their tertiary structure. They also can come together to create a multimeric protein. When they're together, we're looking at their tertiary structure. So, or sorry, quaternary structure. Uh, so tertiary is individual proteins. Quaternary is looking at multiple proteins together that make a complex. All right, so we're going to be in the home stretch in just a moment. Okay, so the final, it's going to come back in a second. There we go. The final kind of interaction that we're going to talk about are hydrophobic interactions. So when it comes to hydrophobic interactions, this is primarily driven by thermodynamics. Particularly, it's going to be driven by entropy. That is the degree of disorder or randomness in the system. And we want to avoid decreasing entropy. We always want to increase entropy. And so, as an example, if we had a beaker full of water, and if we took pentane, a electrostatically neutral molecule, and if we combined it with water, which is not electrostatically neutral, and we mixed it together, what's going to end up happening is that there's going to be pentane molecules present throughout the beaker of water. What ends up happening is that when it's mixed throughout, this is decreasing entropy, decreasing the disorder and randomness, and that's not what we want. It is thermodynamically favorable to increase entropy, increase the disorder and randomness. And so what ends up happening, it is energetically favorable to disrupt and break the hydrogen bonds between adjacent water molecules and to then cordon off or cluster together the pentane molecules. And so then once they're clustered together, that ends up increasing entropy. And so it's this is an example of a hydrophobic interaction where there's something that does not have a charge, that is hydrophobic, and usually what happens with hydrophobic interactions is that they're driven by entropy, and it's usually energetically favorable sometimes to break bonds to be able to separate and cordon off hydrophobic molecules from water or from some sort of aqueous environment. Another really crude example, but I'm sure everyone can appreciate it, is if you combine water and oil in a bottle and shake it up. When you look at it, it's just beads of oil everywhere within that bottle of water. But over time, what's going to end up happening is that it is energetically favorable to separate out the oil from the water. This is all happening because of thermodynamics. This is all happening to try and increase entropy. Okay? There are a couple groups. What ends up, one thing I should note here is that the hydrophobic interactions that occur are some of the most important forces that drive individual protein folding. We talked about separate proteins coming together and how there's electrostatic interactions or ionic bonds that can help determine how they come together. When it's an individual protein, its structure is oftentimes determined by these hydrophobic interactions. When we talk about hydrophobic amino acids, there's one group of the aliphatic amino acids, valine, leucine, and isoleucine. And again, if you look at their side groups, they are electrostatically neutral. And so because of that, they're going to end up being hydrophobic. Valine in particular is going to be important because it's going to be a key amino acid that helps explain sickle cell disease. Another key group is the aromatic side chains, phenylalanine, tyrosine, and tryptophan. tyrosine has, even though it has this hydroxyl group here. Oh, forgive me. There's a hydroxyl group here in tyrosine. What ends up happening is that this is going to be important for cell signaling and phosphorylation events, but they're primarily identified by their aromatic side chains. But the key thing to note is that typically the aromatic and aliphatic amino acids are going to be hydrophobic. And so if we took a moment to try and label all of the positively and negatively charged amino acids and all of the hydrophobic amino acids, what you can appreciate is that many of the positively charged and negatively charged amino acids are kind of littered across the surface and entire structure of hemoglobin. But when we look at these hydrophobic amino acids, they're largely buried internally within hemoglobin. And so, again, if you imagine hemoglobin is found within red blood cells, the inside of a cell is an aqueous environment. It is energetically favorable for these hydrophobic amino acids to be buried internally within hemoglobin, to be away, to have their side chains away from the aqueous environment. And so you're typically going to find the hydrophobic amino acids like isoleucine, valine, leucine, phenylalanine, and tryptophan to be buried within the hemoglobin. Before we jump back into sickle cell disease, I'll stop for a couple questions here. Or I should say, before we apply all of this to sickle cell disease, I'll stop for a couple questions. Is histidine considered hydrophobic at high pHs? I'll get back to you on that. That's a great question. Are there any other questions? All right, so to bring it all together, so what happens in sickle cell disease? There is we talked earlier how it is a monogenic disease. So that means there's one gene that has mutation, and it's beta chain, hemoglobin beta chain. And not only that, but it's one single amino acid substitution. We end up going from a glutamate to a valine. And there's a nomenclature that helps us understand amino acid substitutions. It uses the single letter abbreviation for an amino acid. And so E is the glutamate single letter abbreviation. The 6 here is the amino acid position. So at the 6th amino acid in beta chain, it's usually glutamate. It's substituted for a valine. Can anyone venture a guess why this is problematic based on what we've talked about so far about these amino acids and their properties? Yeah? Yeah, exactly right. Glutamate carries a charge with it, and when we substitute it with a hydrophobic amino acid, particularly a position that's near the surface, we're going to have a hydrophobic amino acid that is available to the aqueous environment. It is not energetically favorable for that amino acid to be there. So something's got to give. So because it's not energetically favorable, it's going to be thermodynamically favorable to break some bonds and move that amino acid. What ends up happening is that the structure of hemoglobin changes to bury valine. As you can imagine, we had a slide earlier that shows all of the different hydrophobic amino acids. When it changes the structure, there's more than one valine. There's more than one hydrophobic amino acid. Now these other hydrophobic amino acids are going to be available to the surrounding environment that are highlighted here in this rotating structure. So now we have a new problem. Other hydrophobic amino acids are accessible to the aqueous environment. So now what ends up happening is we need to bury that now. So what we end up doing, what ends up happening is that multiple hemoglobins will start stacking on top of each other. And if so sorry, let me take one step back. They bury the mutated valine by they bury it and create this fiber-like structure. Now that we have other hydrophobic amino acids available, they're going to be stacked on top of other mutated fiber-like hemoglobins now. And so what ends up happening is that we have within a single red blood cell, we now have these fibers that are forming that from hemoglobin that changes the structure of the entire red blood cell to go from this donut bagel shape that's nice and pliable that can fit through small capillaries. Again, the diameter of a capillary, these really small blood vessels in our body, are usually about the diameter of a single red blood cell. And so it's really important for it to be able to squeeze through. But when it's in the sickled shape, it can't do that anymore. And now these sickled red blood cells that have these hemoglobin fibers within them are going to start stacking on top of each other and creating these occlusions. So the treatments for sickle cell disease. So typically it begins with palliative care. Palliative care is usually helping to manage pain. Patients will typically receive fluids, pain medication, and oxygen therapy. There is also a treatment where a new isoform of hemoglobin has been identified called the gamma chain. It's usually found in fetal tissue and it's important for development. What's been identified is that hydroxyurea actually promotes the expression of the gamma chain of hemoglobin. And so treating patients with hydroxyurea can actually shift the composition of hemoglobin from being 2 alpha, 2 beta to now being 2 alpha, 2 gamma. Patients will also typically receive blood transfusions to receive red blood cells that are not sickled. And finally, another option for treatment is bone marrow transplantation. Red blood cells are derived from the stem cells within our bone marrow. Those are called hematopoietic stem cells. So patients, if they're able to find a matched donor, can actually receive a bone marrow transplantation so that their red blood cells are no longer sickled, but rather can carry the proper I shouldn't say proper, the non-mutated allele of beta chain. Considering that this is a monogenic disease, there's another option that's now available for treating sickle cell disease. Can anyone think of pie in the sky? What's an idea for treating sickle cell disease, given that it's a monogenic disease? Yes. Sorry? Yes, genetic editing, exactly right. So many of you have probably heard of CRISPR. It is an incredibly fast and cheap way to do genetic engineering. We'll actually later in the semester go through how it works as we try and discuss DNA replication and how CRISPR leverages that. But within, let's see, about three or 4 years ago, in one of the biggest clinical journals in the world, New England Journal of Medicine, there was a gene that that was identified to regulate the gamma isoform of hemoglobin, that then was able to help change the expression of hemoglobin and restore red blood cells to a non-sickled condition. That was about I think 3 or 4 years ago. A year ago, December 2023, the FDA just approved CRISPR for sickle cell disease as the first FDA-approved gene therapy. And so there's a lot of opportunities for treating monogenic diseases with gene editing, especially now that it's been finally FDA approved. And so a lot of things can change where, when I took this class 15 years ago, which again, saying that out loud makes me feel a little old, but even though this was a monogenic disease, the treatment opportunities ended at this slide. And so there's a lot of opportunities to try and identify new therapeutic strategies that can help treat these diseases. During the first set of slides, we talked about how the incidence rate is typically 1 in 500 African American births. And I said we'd revisit why. And so sickle cell disease is primarily occurs within African American populations, particularly those of West African descent, because there was an evolutionary advantage to carrying at least one copy of the sickled allele. It typically would confer protection against malaria. And so usually there's oftentimes, if a allele is selected for over time, there's an evolutionary advantage or an evolutionary explanation as to why that's the case. And so in the case of this sickled allele, it was identified about 13 years ago, 14 years ago now, that this sickled hemoglobin allele can protect against the pathogen that promotes malaria. And so that's the main explanation as to why it's been present primarily within West African populations over the years. If structural biology is something you're really interested in, we have a great group of people in that space. And so I'd highly recommend, if this kind of material really spoke with you, I'd highly recommend you check out their websites and see what kind of work they're doing. And I'll continue to try and plug different groups at UVA as well as at other universities. But all of these folks are here at UVA, and I'd highly recommend you reach out to them if this material spoke to you. With that, I'm going to stop the recording, but I'll also be staying for like 15 minutes or so after each lecture for any questions you guys might have. Thanks.